Prof Chen. Ladies and gentlemen, colleagues and friends, good morning. We are delighted to welcome back Professor Eugene Chang following his inspiring lecture at PolyU in 2024. Prof Chen is the Martin Boyer Distinguished Professor at the University of Chicago and a Senior Fellow at Pear. He is internationally recognized as one of the foremost leaders in human microbiome and mucosal biology. Prof Chang's research explores how gut microbes interact with their hosts to shape immunity, metabolism, and overall health. By integrating autobiotic mouse model, advanced bioinformatics, and translational approaches, his laboratory has illuminated how environmental and lifestyle changes reshape the microbiome and contribute to modern disorders such as diabetes, obesity, cancer, and autoimmune diseases. His research has earned global recognitions. In 2025, Professor Chang received the American Gastroenterological Association Distinguished Achievement Award in Basic Science for his groundbreaking discovery in intestinal epithelial transport, the protective role of inducible heat shock proteins, and microbial regulation of host genes. His work has profoundly advanced our understanding of gastrointestinal health and disease. More recently, Professor Chang's group has led the way in study of the fecal microbiota transplants, FMTs. That is the transfer of stool from a healthy donor to our recipient, first approved by the US FDA in the past decades. This procedure has entered clinical practice as a promising therapy for restoring gut health. So, as microbiome-based therapies move closer to mainstream medicines, okay, so Professor Chang's work is very, very important. So you remind us the innovations must always be accompanied by rigor, responsibility, and a deep respect for the ecological and evolutionary principles. So I think without further ado, ladies and gentlemen, please join me in extending our warmest welcome to Professor Eugene Chang.
Thank you, Kai. That was a wonderful introduction. I learned more about myself than I did before. This is really a great honor to be here and to be part of the PAIR program. When I spoke the last time, which was probably a couple years ago, I had talked about some of the tools and technologies that we were developing to really quantitatively assess the state of health of the gut microbiome. And since then, we have been establishing a new program at the University of Chicago called microbiome medicine. And I'm going to talk about that today. So microbiome medicine is based off of the understanding of health and disease through the lens of ecology and evolution. So just as, let me start off with this disclosure. One of the tools that we're developing called GB0001 will be mentioned in my discussion this morning. So what is microbiome medicines? Microbiome medicine is a new discipline in medicine that really addresses this emerging area of healthcare that focuses on health and disease of the gut microbial organ. And the mission statement for our program is to advance the practice of precision medicine by unlocking the potential of the human microbiome to promote health, prevent disease, and personalize health care. So when I think about the gut microbiome, I don't think of it as just thousands or trillions of microbes that live in our gastrointestinal tract. I think of it as a vital organ of the body. It fits the definition If you look in Webster's, for instance, a vital organ is defined as a differentiated structure consisting of cells and tissues that perform specific functions in an organism. So the microbial organ has many similarities to other vital organs or systems in the body. First of all, it provides essential functions. It's recognized as self. Our immune system tolerates it. We cannot live without it. We develop these disorders, for example, Clostridioides difficile, which is something that we see very commonly in patients that are put on broad spectrum antibiotics for extended periods of time. And as was mentioned, it can be transferred. It can be transplanted from one individual to another. And in fact, if the match is not good, many of the microbes will be rejected by the recipient. And like other organ systems, if there's an imbalance or damage to the microbial organ, it can contribute or cause certain disorders. Now, there are differences with other vital organs. First of all, this is an acquired vital organ. It really doesn't depend, and in some cases, doesn't tolerate oxygen. And very importantly, unlike many other organ systems like the brain or the liver or heart, it's easily modified. And that makes it opens up the field in terms of developing future biotherapeutics. And then I want to just point out, because this is something that people don't think about, but the microbiome of the gut is actually several ecosystems or microbes in ecosystems from the stomach down to the colon. And each place is different. And I'm going to come back to this because this really determines the kinds of microbes that live in these regions. So why are gut microbes so important? Well, I think most of you in this audience know this. It's been shown to be vital for regulating gene expression and function of the various parts of the GI tract and also GI tract associated organs like the liver. But we also know that has direct modifiable functions on the brain, heart, liver, and other vital organ systems. And when things go awry, many disorders can arise. Some are shown here. So you can see that the impact of an imbalanced or damaged gut microbiome can affect not only digestive processes, but also processes that are extraintestinal. So as I mentioned, medicine has many specialties. They're shown here. This is certainly not an inclusive list. But I think that what I'm going to talk about today is what I think will be the next specialty in medicine, and that's microbiome medicine. And at the University of Chicago, we have been thinking about, well, how do you develop this area of microbiome medicine as a medical specialty? Well, first, it has to start with developing clinically useful diagnostics and management tools and technologies. It's like any other organ system. This has happened over decades of time. We have imaging studies, laboratory tests, and we have other types of metrics that we use to assess the state of our vital organs. We don't have that right now for the microbial organ. Once we have the ability to measure the health or unhealth of the gut microbiome, this opens up opportunities to develop biomarkers and predictive AI platforms that can not only tell you whether a person has a particular condition or disease, but can predict it. And if you can predict or identify individuals at risk who will develop a microbiome-related disorder, then you are shifting the paradigm in medicine. because medicine today is really reactive. We wait till the patient develops disease, and then we try to treat it. The outcomes are not always great, particularly for chronic disorders. If we could shift this paradigm to prevention, that is, which requires identification of individuals at risk, that's where we want to intervene. That's where we want to try to change the natural history so that we can mitigate or prevent the development of a condition or disorder. This also has to be precision. You know, today's goal is trying to make interventions and therapies more precise so that they're targeted to the particular needs of an individual. And then I think we have to develop means of studying the microbiomes that are outside of the colon. For example, the small intestine is no man's land. It's essentially understudied because we don't have the tools and technology to be able to sample the small intestine in a way that we can do in the colon. Okay, so let me begin by saying that, you know, if you look at all other vital organs of our body, we can define normal by metrics that we have developed. And this is true of the five vital organs shown on the right. But all of these have reference ranges. We know what is normal. And just as an example is a screenshot of my blood studies. And you can see that normal is defined by where you are in that green zone. If you're outside the green zone and in the yellow zone, then physicians know that something needs to be done to find out why. Okay, so this is the cornerstone of medicine. Unfortunately, for the microbial organ, we really have nothing. Okay, so this is what my group has been working on for the past 5, even 10 years, and thinking about it and actually developing new tools and technologies that can be used clinically at a price point that's going to be acceptable to the patient. So what do we have today? Okay, so many of you know that we have this technology that has been very useful, where we basically can profile a single gene called 16S ribosomal RNA. And the sort of polymorphisms, the differences in various regions of the 16S RNA, ribosomal RNA gene provide us a way to identify a particular organism. Now, we can't get down beyond a genus level, but it gives us a taxonomical profile. So it tells us who is there. And this has really been used now, today, by most academic and commercial groups. The reason why is it's fast, cheap, and it's easy to determine who is there. Unfortunately, its limitation is that it doesn't tell you what they are doing, okay? And I would argue that's much more important for research as well as for clinical care, because it really doesn't matter who is doing it. You want to know what they're doing. So we have shifted to functional markers. Now, one of the functional markers that has been used more oftenly now is called shotgun metagenomics. So this is where you go in and you sequence all the DNA in a particular sample. And here we have found, as you can see in the lower panel, data from the NIH Human Microbiome Project. So they had over 1,000 subjects, all healthy. Okay, and their results are pretty much stacked along the x-axis. So you can see that from the left to the right, the colors are pretty uniform. That tells us that we as a human race pretty much have microbes that share the same or have the same kind of genes. And to me, that has implications because, again, it's not who is making it, it's what they're making that's very important. Okay. So can we use shotgun metagenomics clinically? The answer is probably not. This is really a research tool. It takes time to do the analysis. The other problem is that probably 98% of the features or the various peaks that we can see, we don't know what they are. So it's what we call dark matter. And most of us stay away from the dark matter. And we look at the 350 known knowns that we can identify. But even that is a very limited amount of information. So my lab has shifted towards metabolomics. Metagenomics is also limited in the fact that it tells you how many and what kind of genes are present in a particular sample. But it doesn't tell you which of those genes are actually being expressed and to what extent. So it's an indirect measure of gut microbial function. Metabolomics, on the other hand, is a direct measure of microbiome function. And microbiome here is defined as not just the microbes, but the microbes in that particular regional ecosystem of the gut that they're in. Okay, so this is a composite measure, but it is a very good measure because it's a direct measure of function. Now, we can measure this, as I mentioned, using an instrument called mass spectrometry. Now, the good thing about mass spec is that it's a technology that is widely used. It gives us a lot of information. The problem with mass spec is that it gives you too much information. So you get 10,000 features, for example. Now, for somebody to sort through all those 10,000 features is just a nightmare, right? And it's not going to be clinically useful because we don't know what 98% of those features are. So what we decided to do is take a different tact. We took those 10,000 features and we subjected it to a machine learning and AI supervised filtering. And we had two major criteria. The first criteria was that whatever we were looking at, we had to go with known knowns, because if we didn't know what the feature was, then we could never establish a reference range for it, right? So we looked at the 350 known knowns and we asked, well, which ones are most homogeneous in a healthy population? That's a requirement if you're going to try to establish normal reference ranges. So doing that, we were able to maybe get 50 that had fairly good reference ranges. We then asked, well, of those 50, which one best represents a specific functional subsystem that a healthy gut microbiome must have? For example, fermentation, amino acid biosynthesis, bile acid conversion. So these are categories. They're pretty much like the warning lights that you see on the dashboard of your car. If your tires are low in pressure, your engine getting too hot, whether or not you have other problems that need to be looked at. So this is what we developed. And we came down to 20 markers that performed not only well individually, but they performed very well together. And because of that, we developed something called the Z-score. So the Z-score was basically the sum of the deviations of a person's value from the normal range of that particular marker. And so if you had a Z-score over 20, we define that as a dysbiosis. If you had a Z-score of less than 15 or 20, we define that as a eubiosis. Okay. So this is actually a quantitative way of assessing, defining a healthy or unhealthy gut microbiome. And it's first of its kind. The other thing is that because we know with these 20 markers, we can identify specific problems that are driving a dysbiosis. Okay. All right. So is it useful? Okay. This is one of the cases that I frequently show, because I think this was the first case that showed we actually had developed something that can be diagnostic. So this is a patient that contacted me now about 15 years ago. And at that time, he had a 10-year history of suffering following 18 months of antibiotics that his doctor put him on. And he had symptoms that included incontinence, stomach pain, food restriction. He couldn't tolerate many types of food. And he had chronic GI discomfort to the point where his quality of life was really very bad. He had gone to 4 major academic centers in the United States. They could not find anything. All his blood studies were normal, and he had a normal colonoscopy. So when he contacted me, he asked me, might there be something wrong with my microbiome? And I told him that I don't know, but I have a test that might be able to give us that answer. So the answer is that he had a very severe dysbiosis. If you look at panel B, so before we started intervention, he had a Z-score of 42. That's really abnormal, because these values are in log units. So we knew he had a dysbiosis, and then we looked at the individual functional subsystems. And what was driving it was the fact that he had very almost undetectable levels of things like short-chain fatty acids. These are fermentation products that are produced by the gut microbes from a substrate of dietary polysaccharide. But he had other problems. Now, part of this was because he was on such a Spartan diet. And so we asked the question, well, is it because of the diet? Maybe the microbes are still there, but he's not feeding them, right? So we can't detect them. We can't detect the products they're making. So we put him on a intervention that basically would introduce various food groups over time. And what you can see is that with the panel that we developed, he progressively showed improvement of his microbiome health, to the point of after 41 weeks, he actually had a eubiosis. So he had a Z-score of less than 5. And at that point, he was feeling much better. He could eat diverse foods. But without this management tool, I don't think we would have been able to really manage him this precisely and effectively. Okay, so I'm going to skip to the other part, and that is that, okay, we developed this tool to assess the state of health of the gut microbiome of healthy adults. Now, it turns out the adult microbiome actually starts around age of 3 or 4, and then remains very stable to about the age of 70 or 80. And during that time, or outside of that bracket, the microbiome is changing. For example, in the first 3 years of life, we know the microbiome is changing, it's maturing. And then after about 70 or 80, many people show a progressive decline in their microbiome, even though they're healthy. Okay. So we couldn't just use reference ranges. We had to look at a different way of measuring health of the early and late life microbiome. So we decided to measure normal trajectory ranges. So this is like what pediatricians use, for height and weight. It's very, very useful. And what we're doing now is we're informing these platforms and developing reference ranges, both in early and late life, such that if you look at the panel on the left, if there was an individual, an infant, that was off trajectory, that individual would be maybe benefited by an intervention at this point. And the object would be to drive this back to the normal range. And by doing so, maybe you can lower the risk of many disorders, particularly neurodevelopmental disorders that include autism spectrum disorder. Okay. Now, why that's important is that once ASD develops, we only have symptomatic measures. We can't really treat or cure ASD. The damage has been done. So the goal is, in this case, for example, to identify those kids within the first year of life that are off trajectory, and then introducing interventions that will change the course through restoring the health of their gut microbiome. Okay. All right. So I told you that, a lot of people don't look at the dark matter. And I think that we avoided the dark matter for a long time. But as I began to think about it, the dark matter has a lot of information. And maybe we can use it, but we approach it from a very different way. Instead of developing reference ranges, let's look at signatures. This is just like facial recognition or fingerprint analysis, right? You're not looking for absolute values. You're looking for patterns. And this is in the figures below is a mock-up of some of these types of panels that we might see. For example, healthy may look like what we see in A. And let's say a patient that develops C. diff, where you're wiping out most of the microbes, you see very few features. And then other instances are like IBD and prodromal dementia. Now, does this work? The short answer is it does. So we took the database that came out of the Broad Institute for IBD. So they had a fair amount of data that was gotten from patients that were controls, had active IBD, and then they even separated them into Crohn's disease and ulcerative colitis. So we had a very large database to look at. And we looked for these signatures. And it turns out that IBD versus control was easy. The patterns were very, very different. But we could also discriminate with very high performance, AUCs of greater than 0.9 between Crohn's disease and ulcerative colitis. Okay, now we're working on this further because we can refine this technique in many different ways. And which I don't have the time to get into today. But I think that the dark matter, unlike GB1, it becomes a way that we can use it as a diagnostic tool with high specificity. Okay, so I'm going to switch gears now. I've talked up to this point about, that we have to develop tools that are diagnostic or predictive. And we're doing that. And we have a lot of data that would suggest that the approaches that we're using actually do work. They still need some refinement. Well, what about going forward? I mean, now as we're developing these tools, it brings the opportunity of thinking about how we can develop the future precision interventions to correct a dysbiosis or to maintain a healthy eubiosis. And the goals here would be twofold. One is that you have to be able to restore the health of the gut microbiome. And that's below. Okay, so we're doing that already. A lot of probiotics, prebiotics, postbiotics. We're using this in a way, but now we have a tool to actually determine what these types of interventions that will change the microbiome are doing, and whether or not it is customized to that particular individual. But the other part of it is the host side. You have to be able to restore gut homeostasis. And I'm going to give you an example of that shortly. But this is very, very important, because if the ecosystem is damaged, you're never going to engraft a healthy eubiosis. So what do we have now to maintain a gut or restore a dysbiosis to a state of eubiosis? Well, we have prebiotics, which I actually think work fairly well. And I think that this is a vision that, for example, Rye Foods has as well. And I think that there's a lot of areas that of common interest I'd like to explore further with the Rye Food group. They're live biotherapeutic products. These include things like probiotics, genetically modified microbes, phage, FMTs, and what I call omni-microbial transplants, because omni includes small and large bowel microbial transplant. And I'll tell you why that's important in a few slides. And then there's postbiotics, which actually I think make a lot of sense and probably are very effective, because now you don't have to depend on engrafting the probiotic or the FMT. You give what they make. And this is a way that you can control things much better. Okay, so there are a few ecological and evolutionary principles that have to be taken into account when we're developing new microbiome-based interventions. And I think the first thing is that, and this was shown very nicely by one of the research scientists in my group, Dr. Na-Fei. And what she did was that she asked the question, well, can you just engraft any type of microbiome, let's say from a fish or a bird, or from a mouse into, let's say, a human being, or can you take human being microbiome, put it into a germ-free mouse? And when she asked the latter question, it was very clear that there's a selectivity among hosts for their particular microbes. And so what we're showing in both panel A and B, on the left is observed OTUs, on the right is the evenness. And you can see that in very stark differences, that when you put a human microbiome into a mouse, less than 50% will engraft. Now that gives concern about all this literature that is being used to study and characterize human microbiomes in a germ-free mouse. Because what's happening is that you're skewing the population. If 60% of your human microbes never engraft, then you're only looking at the function of the 40%, right? So we have to think about that. And the other thing that she found was, she went through everything. What drives this, what we call phylosymbiosis? This is where we think that the gut microbiome has co-evolved or co-speciated. But we wanted to find out, was it immunity? Was it mucus? Was it the diet? And all these things. And we went through an exhaustive list of things, and nothing explained the phylosymbiosis except for this. And this is basically a relationship where we're looking at phylogenetic distance between species from the mouse and the percent of engraftment of FMT from those species into a germ-free mouse. And you can see that the farther you are away from the mouse, the less engraftment you will get. So there's a linear relationship. So what actually drives phylosymbiosis? I don't know. But what it tells me is that non-human derived probiotics engraft very poorly in a human gut ecosystem. So if you're going to develop a future probiotic, it's best to get it from a human. Okay. And that's something that even industry either does not recognize or chooses not to recognize it. Okay. So the other ecological principle is that you have to have the right microbes in the right place. And this was a study that we did in collaboration with investigators at the Chinese University, Hong Kong. And what they did was that they had a study where they administer human FMT from above, and then 4 weeks later would take samples from the duodenum and also biopsies. And what we found from those samples was that there was an increased anaerobic engraftment, a lot of anaerobes in the duodenum 4 weeks later that aren't supposed to be there. And you have to wonder why. A lot of these anaerobes are strict anaerobes. So the answer was provided by a former postdoc, now part of our faculty as a research assistant professor, Orlando De Leon. And what he found was that, or what he did was he took either jejunal microbial transplant into a germ-free mouse, or that mouse was given a FMT. And what he found was that if you get the wrong microbes in the wrong place, as shown in the bottom here, basically, this is normal up here, where there's matching of jejunal microbes in the jejunum and colonic microbes in the colon. But if you get a mismatch, which happens, let's say if you give JMT, a lot of these facultative small bowel microbiota engraft into the colon. And vice versa. If you give FMT, a lot of anaerobes, strict anaerobes, will now engraft into the small intestine. Now, why? These are not the right places. The small intestine has oxygen. A lot of the colonic microbes cannot tolerate oxygen. Colon is primarily anaerobic, and facultative organisms cannot live in that environment. So what happens is that when you have a mismatch, the microbiota that's out of place will terraform the regional ecosystem. So what we found, or what Landon found, was that you put jejunal microbes in the colon, and the colon begins to change and takes on some of the small bowel identity. And this is by transforming some of the stem cells. And vice versa, if you put FMT down the small intestine, you begin to transform it into a colonic identity. And this is why the wrong microbes can now stably engraft. For example, if you put FMT into, gavage it into a germ-free mouse, that small intestine becomes more colon-like. You see increases in oxidative phosphorylation, which increases in the anaerobic environment. And the consequence is that now these microbes can engraft. Okay. So if we're going to develop interventions, we got to get the microbes into the right place, right? All right. Now this happens actually in real life. Okay, and this is a study we did as part of the Human Microbiome Project years ago, but we identified through this human model where patients with ulcerative colitis would have their colon removed, and then they would have the distal ileum fashioned into a pseudo-rectum so that patients would have continence. And what happens over time is that pouch, the ileal pouch, gets colonized by colonic-like microbiota, probably because of the stasis. And it develops an anaerobic environment. And the reason for that is shown here. What we're doing is that we have now colonic microbes, or we have the ileal pouch or mucosa now being exposed to a colonic microbiota. So we use two markers, for example, FAB-B6, which is an ileal marker, and carbonic anhydrase 2, which is a colonic marker. And you can see that in the pre-pouch, that's the part above that is fine, it doesn't get inflamed. You can see that these markers are very good for small and large bowel mucosa. In contrast, if you look at the pouch, after 4 months, you can see staining of for example, FAB6 in here. You see a bitty patchiness here. And with carbonic anhydrase 2, you can see that it's really, this small intestine is not only exhibiting gene expression that would be typically seen in the colon, but you see now that the kind of villus-like structure that you typically see in a normal ileum are now gone. And now you see these crypts. This looks like a colon. Now, very quickly, all of these changes, at least the genes that we looked at, occurred through epigenetic modification. Now, whether it's reversible or not is something that we're trying to investigate at this point. Okay. All right, so I want to tell you now another story. And this one I think is really important. And this is why I truly believe that if you have to understand the ecology and evolution of that underpins host microbe interactions. So this was done by a brilliant graduate student of mine, Megan Kennedy, who did an experiment to ask the question, well, how do you repair a damaged microbiome? So she damaged the mouse microbiome using a combination of Western diet for 3 weeks, and then that was followed by a cocktail of antibiotics for over 3 days. And as you can see above, what happens is that if you maintain the mice on a Western diet, you really never recover. You have a microbiome that remains less diverse, and the metabolome changes, as well as the metagenome. And even by 63 days, you can see that it doesn't look like anything before the antibiotics were started. Now, on the other hand, if you put these mice on a mouse chow, which is high fiber, low fat diet, you see very rapid recovery after the dysbiosis is induced. And it occurs in what we call a successional pattern. It's not random. And what it's characterized by is essentially shown here. There's a lot of data I'm not showing you here, but basically we got a lot of metabolomic samples over time, and then we fed it into a computer model program. And what explains the successional pattern in, let's say, when you put them back on a high fiber, low fat diet, is that you get almost immediate return of these keystone or foundational species. And they're producing metabolites that basically set the stage for the next succession. And then that group produces even more diverse nutrients and conditions so that it sets the stage for ultimately the return of a nearly normal microbiome. In contrast, if you maintain a damaged microbiome on a Western diet, it never recovers. These organisms that are appearing first, they're not keystone or foundational. They are organisms that pretty much have genomes where they can take care of themselves. They don't care about the other members that would potentially be where they could be interdependent. They're very independent. So it really never recovers. And I think this is important because to me, diet is underappreciated in terms of modifying the gut microbiome and in restoring conditions that may lead to disorders. Okay, so this is shown in a different way. Okay, so you probably have been reading the literature. A lot of people are using FMT to treat patients with inflammatory bowel diseases. Now, does that make sense? Well, so if you take a healthy community and you put it on top of a inflamed gut, it's not going to engraft. Okay, there's no chance that most of those organisms will disappear. And so this is a point where you have to stage your interventions in a way to recognize these ecological principles. You have to restore the ecosystem before you start rebuilding. Okay, and to do that, we, for example, would use immunobiologics or anti-inflammatory agents that try to get the ecosystem back to a state where now you can think about bringing the succession of microbes that can restore, rebuild that microbiome. So eventually you can reestablish eubiosis and keep that patient in remission, for example. Okay, so what are the paths for restoring gut microbial health? Well, there are a few things that we have to recognize. First of all, one size will not fit all. All right, there's not going to be one therapy that works for everybody. Okay. The second thing is that host dietary microbiota factors all have to be taken into consideration. Right now, our treatments are basically only focused on the microbial compartment, but we have to think about the host side as well. Diet is very important. I personally think that diet is the best way to treat dysbiosis and to maintain a eubiosis. But there's something else that we have to recognize. It's not just who is there. We also have to know whether or not they are playing well together. And so one of the things that we can do is we can measure stability and resilience. These are properties of a healthy microbiome. And I think that this is something that should be incorporated not only in the sort of diagnostic part of the of microbiome medicine, but it also has to be one of the properties that we try to restore or maintain in patients that have a dysbiosis. And as I showed with the IBD example, the type of and timing for interventions is very critical, and the method of delivery is very important. So I'm going to summarize this. I mean, this is obvious, but it pretty much follows the seed and soil principle. We have to keep this in mind. Good seeds in a rich soil is going to produce good outcomes. Bad seeds in a bad soil is going to remain unhealthy. You put good seeds into poor soil, like in active inflammation, it's not going to graft and it will continue to be unhealthy. You put bad seeds into a good soil, it's also going to be unhealthy. So I think that all of these things, as simple as they sound, are really, really important. Okay. I'm going to end with this, and I mentioned this earlier, and that is the future of microbiome medicine, I think, lies in prevention. And this is a very famous individual, one of the founding fathers of the United States, Benjamin Franklin, who said, an ounce of prevention is worth a pound of cure. I'm a true believer in that. And so thinking of the future, we're developing these platforms. These are what we call large science models, or LSMs. And this was from a grant that we submitted to identify infants at risk in the first year of life for development of autism spectrum disorder. And basically, it involves the LSM is basically down here. And we have to have input data, this high quality input data to train the AI. And this has to be longitudinally collected, but so we know what the outcome is. And once we train the AI, we can then create what we call a digital twin. This is basically an in silico model of that individual. And that becomes very powerful because now it's like forecasting weather. If we can input clinical metadata, laboratory studies, multi-omic studies over time, and we've seen the progression of this in the past in many individuals that go on to develop, let's say, ASD or diabetes, IBD, it becomes a forecasting tool, right? And this isn't just speculation anymore. This actually does work. It just takes years to develop it. But if you can develop it, then you will have a paradigm shift in medicine. Now you're going to be able to identify individuals at risk before they develop the problem. And at that point, you can do in silico modeling with this AI platform to be able to see what combinations of interventions are best for this individual to change that natural history and de-risk that individual. So that is the future. We're not there yet, but these are things to consider. So I'm going to end here. A lot of people went into this. This is not just me. I'm probably one of the minor people, but the ones that are bolded here are the heavy lifters for developing tools and technologies and thinking about what the future of microbiome medicine is. So let me end there, and I'd be happy to answer any questions. Thank you.
Thank you for Professor Chang for a very inspiring talk. You know, even it's not the first time I attended Beach, but every time I could learn something new. So it's good that we have quite a few audience on site today. So first of all, I would like to invite if we have any questions from the floor to the speaker, Professor Chang. Yes.
Thanks so much for your wonderful talk. I'm Jacqueline Sim from the School of Nursing. If I can, I have two questions. One is for personal purpose and another is for clinical reason. For personal purposes, a lot of the non-prebiotic and postbiotic food are dairy products. So for people who can't tolerate or can't take dairy products, what you would suggest as a healthy alternatives?
Well, so I'm a skeptic of commercially available probiotics. Claims are made with no scientific evidence. So to me, that until scientific evidence is shown, I don't think, I'm guessing most of them don't engraft, don't do what they say it does, and it passes right through you. But I think we can be smarter. We can design better prebiotics. Postbiotics, I think, do have health benefit. You know, kimchi, other types of fermented foods. This is where the microbes are already processed and produce the nutrients that will benefit us. So I think that probiotics are maybe the most effective way, because you can develop postbiotics in a way that doesn't require a microbiome to make them. Okay. So what was your second question?
Can I ask the second question? Yes. I work with people with autism. So for us, or for me, as a mental health nurse, we see it as a neurodevelopmental disease. Quite likely is the person already born with it, but we started seeing signs and symptoms. But I was very interested in you showing the slides talking about the microbe, and I'm sure you're aware of the controversy and the paper reporting false data about the MMR combined jab on children and suggest that that might have caused autism, and that still have a lasting legacy on parents not letting their kids have the jabs. And what do you think about that?
Well, first of all, most association studies have to be validated. I think it's easy to find associations, particularly with the sort of recognition platforms that we have. But it doesn't mean that when you find an association, there's cause. And more often it's an effect or an epiphenomena. Okay, so I'm a hardliner that you have to have scientific evidence. Now, you're right that there's many things may cause or predispose risk for autism. I happen to believe that gut microbes play a very important role. We know, for example, that in a notobiotic mouse, a germ-free mouse, if you do an MRI on that mouse compared to a mouse that has a healthy microbiome, the developmental patterns are completely different, and they occur at different times. And what this tells me is as your microbiome is developing, it's producing metabolites that are essential for development of specific parts of your brain. And brain development does not occur all at once and all at the same time. It occurs in different stages. So you have to have the right mediators or stimuli and conditioning factors at that right time. So it's complicated. Now you say that many kids are born with risk or maybe latent autism. I agree with that. So this is why I think that we have to look at maternal microbiomes, particularly in the third trimester. Now, the other reason I'm into microbiome and ASD is the fact that I said very early, the microbiome is very modifiable. Okay, a lot of other things, genetics, are not easily modified. So it's something that we can do. So if we look at the mother's microbiome at the third trimester, which is vertically transferred to the infant, if we can modify that in a way that the vertical transfer of a eubiosis rather than a dysbiosis, I think that we can make a difference in the incidence of autism.
Hello, President. I think this is the first and most inspiring talk I've heard so far on microbiome. I love your take on the ecology and the evolutionary perspectives on it. And because I'm in clinical transformation work, so I'm working as the chief of clinical transformation at Virtus. So what I'm very interested in knowing is what is a critical point to determine whether the ecosystem has been established for microbiomes? Because this will then set the stage for all referencing benchmarks. Okay, so that's the first question I have. The second is we have microbiomes in the market commercialized right now for the main purpose of preventive health. But then what you're telling me in the actual presentation here, if I am correct, is that most of the post probiotics is actually more ad hoc nature. So they're more interventional than preventive health. So I want to know what your outtake on that is. And also, is there a critical age in which these microbiome ecosystem change or evolve significantly enough to say, okay, from this point onward, you need to do a next cohort or next age corrected cohort to establish these benchmarks because this will invalidate the results that you obtain. So these are my three questions. Thank you.
So all three questions are excellent. And unfortunately, I don't have the answers because this is such a new field, right? And we're still crawling. And you have to learn how to crawl, walk, and then run. So we're not even close to the walking and running. So in terms of, how do we change an ecosystem? Well, actually, let me go back. So rather than looking at how to treat a dysbiosis, I think the emphasis should be on how to maintain a eubiosis. Okay, we can do that already using the tool that we develop because we can tell. We've done several dietary studies, for example, in neighborhoods around the University of Chicago, and they're teaching good, healthy cooking and healthy food choices. And so we sampled them before and after 8 weeks on these classes, and you could see measurable changes in those patients that had a dysbiosis. They moved towards eubiosis. Those that were eubiotic maintained eubiosis. So there we had evidence that that program was actually very effective. Now you can imagine that this could be applied to whatever food products or supplements that you develop, because now you have a quantitative measure to determine whether or not you can maintain eubiosis or actually restore a dysbiosis to eubiotic state. On the host side, how do we sample the ecosystem and determine how that can be fixed if there's something wrong is more difficult. It's limited by access. That is, we can sample by colonoscopy the colon, but the small intestine right now is like, we don't have any instrumentation to sample the small intestine. Now we're trying something new. We're working with a company that has developed these smart capsules, and the smart capsules will open after passing the stomach, and it will collect DNA, metabolites, and also live organisms. Okay, so it will, and it closes before it gets into the colon. And so our goal here is to see what the healthy small intestinal microbiome looks like. And then we also want to develop a library of live biotherapeutic products from the small intestine, so that in the future, if we find that somebody has a small bowel dysbiosis, we have a way of treating it.
I see that. You said that you will be working on a project on autism, right? So I can see that pediatric sort of outtake and preference, right, for working on microbiomes. What's your take on working with elderly population? Because a lot of microbiomes, probiotic stuff are literally being sold to people who are 50 plus right now in the market.
Yeah, so I can probably answer that in about 5 years. Okay, because we just got an NIH grant to study that, right? So the tools that we're going to be using are both the known knowns and also the dark matter approach. And what we're going to try to do is identify individuals that are in what we call the prodromal phase of either dementia or sarcopenia or frailty. These are age-related complications. And I anticipate that people at risk for those complications are going to show a much an off trajectory decline in their microbiome health. And it's at that point that we want to intervene. And there are many ways that we can intervene. It could be probably diet, probably is the best way, but there might be in the future live biotherapeutics that we can develop.
Thank you. I think we have the last questions from the floor. Yeah. Maybe the next one.
Yeah, thank you, Professor Chang, for this wonderful talk this morning. I'm Ravindra Karki, a second-year PhD student here at Professor Chow's lab, and I've just begun my research on development of probiotics for Candida albicans infection prevention. So my question here is, if I understood this talk well, so a healthy gut microbiome translates into a metabolite profile that is defined by 20 key metabolites, I suppose. So is it possible? So it implies to me that these metabolites also help in turn to sustain this healthy gut microbial community. So instead of doing fecal microbial transplant, now can we manipulate like metabolites and bring back the dysbiotic microbiota profile back to eubiosis? Is it possible? Thank you.
I think it is possible. There are some commercial products already out there. For example, short chain fatty acids, which are usually never, if you ingest them, they're rapidly absorbed by the small intestine, but they have synthesized polymers of, let's say, butyrate. And that has advantages. Butyrate smells very bad, right? So a lot of patients don't want to take butyrate, but if you make it as a polymer, the linkage is where it's hydrolyzed by colonic microbes. So you can ingest it, and then it will be put, the monomers of the short chain fatty acids will emerge. So that's an example of how you can design a drug to go in the right place that is a postbiotic. Okay.
Sorry, due to limitation of time, we probably leave the last questions here. Yeah.
You can come down
Yeah, maybe we can have
and
a
I'd be happy to
conversation
discuss
after
it.
the whole seminar. Yeah,
Oh,
thank you.
okay, Prof Eugene, it's a very good talk. I just have two short questions considering time. I thinking you already mentioned about this gut microbiome is kind of like, it's not only, I believe it's black material, also it's black box. So you highlight with a large language model, but the point through metagenomic, so we only know who's there and how they function, really have this kind of the whole complex process. Is still a lot of way to go. But even there are not another like cultural method, we only less than 1%. So the whole black box in this large language model, can we solve the problem? Yeah.
So the short answer is yes, we can solve it.
Okay.
Okay. For example, metabolomics, you get 10,000 features, okay, and 98% are dark matter. Okay, so what we use is a different instrument, a different mass spec instrument. We use an Orbitrap. Okay, so Orbitrap gives you very high resolution. The more it goes around, the higher and higher the resolution. And it's very cheap. Okay, so at the University of Chicago, to rent the Orbitrap for one day is $200 US. You can run probably 200 samples in that day. So that's $1 a sample. And you get very high resolution of these patterns. And I think that these patterns can actually be used to be disease, that will be disease specific. Okay.
Okay, thank you. My last question is, consider we have evidence to show there's connection between gut microbiome and other organ, and particular like a neural system as well.
Yeah.
So what's the best way how we can classify there's connection through the microbiome, through other signals or whatever we're going to push for the next wave or stage for research?
Yeah, so that's an area that's actively being investigated by numerous groups, you know, the brain gut or brain the gut microbiome brain axis. It's thought to be important for not only health but also disease. But I believe that the microbiome does impact the brain. We've seen it with the MRI of developing mice. But on the other hand, we have seen it when you have a mismatch, like what Dr. DeLeon had reported. So he's done studies now looking at a single FMT given and engrafted into the small intestine. He sampled the brain, okay, a particular part of the brain that controls circadian rhythms called the suprachiasmatic nucleus. That's where the optic nerves cross. And he looked at gene expression. A year after a single FMT, the gene expression in that SCN was still abnormal. So people assume that FMT is safe. I worry that we will begin to see, as it being used more and more, short and long-term consequences. Whether that's reversible or not, I don't know. That's something we're trying to figure out now.
We actually, thank you, Professor Chang. We actually received quite a lot of questions from online, but I noticed that we have a few colleagues from social science, School of Nursing. So maybe I choose the one that is more relevant to that. So the question is, how does microbiome medicine change the way we provide traditional healthcare and preventive healthcare? What's your opinion on this?
Well, that's going to be the major focus of our what we're doing now and in the future. I'm a big believer in prevention. And prevention is always going to lead to better clinical outcomes than treatment. Always.
You also mentioned the ability of grafting the microbiome from different species, actually, which is highly depending on the response on the host, right? So there is a question here that's related to that. How would gut microbiome benefit from how would the gut microbiome benefit in combating antimicrobial resistance and improve immunity in immune-suppressed patients?
Well, that's really an excellent question because I think that is a major challenge in medicine now. We have, for example, a lot of patients that undergo bone marrow transplant. They develop, they're put on antibiotics and then they develop these antibiotic resistant organisms. And there's just no way to treat them. But it turns out that probably the best way to treat them is by preventing the emergence of these organisms. And you can prevent it by trying to restore the eubiosis as quickly as possible, or maintaining a eubiosis or FMT. Okay, so this has been shown by several investigators, but the best investigators that has done this was Eric Paymer at the University of Chicago, who showed in a clinical study that if you can give a defined FMT, you can restore pathogen resistance. So these antimicrobial or antibiotic resistant organisms basically take advantage of, because they have no competition. But if you put the competition back, they will disappear. They will never engraft.
Okay.
So,
okay, thank you. I think we still have a bit of time. So we have talked about the neuron-related disease such as autism, right? But how about for other neurodegenerative disease like Parkinson's disease? Do you think microbiome medicine should prioritize static microbial signatures or dynamic response phenotypes?
Okay, so this is the NIH grant that we just got. Okay, so I don't have the answer. We haven't even started the study.
Okay.
But the hope and anticipation is that we will identify microbiome signatures that are associated with the development of alpha-synuclein, which is this protein that becomes malformed and is believed to be important to the etiopathogenesis of Parkinson's disease. And there was a very good study by a very good investigator that thinks that the alpha-synuclein pathology begins in the gut. And it begins in either the neurons or some stromal cells of the gut. And then the alpha-synuclein travels up the vagus nerve and then will cause the development of Parkinson's disease. And so the hope is that if we can identify people that have a eubiosis that's driving, let's say, gut alpha-synuclein production, we can mitigate the risk for development of Parkinson's. But we're just, so this is a 5-center study. Mass General, Mayo Clinic, us, Rush Medical Center, and Stanford. And we just began, and we're collecting samples of patients that are prodromal, just very early development to see what markers they have in the microbiome that might be associated and maybe contributing to the development of Parkinson's.
Okay, thank you. Well, just a bit forecast for the seminar. I think it's about times. We will have a seminar hosted by iFood this Thursdays. So Professor Chang will give another talk, which is also microbiome oriented but different focus. So like if you're available, please, you are all welcome to come. Okay, so I think that's about it for today. So we would like to present the souvenir by Professor Wang to Professor Chen.